All right. It is exactly noon, and we are ready to start. We have two speakers today, Dr. Bernado and Dr. Rangachari. The talks will be half an hour, give or take, followed by about 10-15 minutes Q&As after each talk. And at the end, we will have a discussion in which all the attendees can address their questions to both speakers. And the first speaker, it is my pleasure to introduce Dr. Bernado, who is a professor at the Center de Biologie Structurelle in Montpellier, France. Dr. Bernado obtained his PhD at the University of Barcelona in 2003, where he combined NMR and computational methods. Then he moved to Grenoble for his postdoctoral studies in the lab of Dr. Blackledge, and during this period, he worked on molecular modeling of disordered proteins using NMR data. Then he visited EMBL Grenoble in the group of Dr. Dimitri Svergun, where he learned small angle X-ray scattering and developed approaches for ensemble interpretation of SAX data. His current research interests focus on the structure-function relationship of disordered proteins and combines NMR SAX and computational approaches. Very nice to have you today, and looking forward to your talk.
Thank you very much, Magda, for the introduction, and thank you, Rams, for the invitation to come to this ZOOMinar. I'm going to present today the work that we have been doing for the last 5-6 years in the Centre Biochimie Structurale in Montpellier, and it's related with Huntingtin and Huntington's disease. I mean, probably many of you know about this disease. Huntington is a neurodegenerative pathology that is characterized by the presence of large cytoplasmatic and nuclear inclusions that causes neuronal degeneration. As a symptoms, the most obvious one is the involuntary movements, the general motor impairment, disorders, the dementia, etc. Normally, this disease appears at adult ages, and death occurs after 10 or 15 years after the onset. It's interesting to see how this disease was described for the first time in detail by Professor Dr. George Huntington. And he says something like the name Korean is given to the disease to account for the dancing properties of those who are affected. And it is a very appropriate designation. So Korea means dancing, and in reality, this is the most obvious thing that patients do. And the will of those patients is there, but there is the way to perform is deficient. It says that it seems that there exists somebody that is hidden with a hidden power, something that is playing tricks to the people. So this is what normally is defined as chorea, and after this description, this disease was called Huntington's chorea, and or also more known as Huntington's disease. So in terms of onset, in terms of hallmark of amyloid, let's say, Huntington is linked to pathologies such as Alzheimer's or Parkinson. But if we think in terms of genetics, Huntington disease belongs to a larger family of diseases that are called the trinucleotide repeat disorders. Those disorders occur when some small fragments of DNA, which are already repeated inside of certain genes, unrelated genes, are repeated even more in an abnormal number, let's say, and this causes the disease. When those expansions occur in non-translated regions, then the RNA that is produced, let's say, this is a toxic species. However, when those expansions occur in exons, as in the case of Huntingtin, then these expansions are translated into amino acids. Concretely, we are dealing with Huntington disease, which is a polyglutamine related disorder, because those expansions of CAG trinucleotide induce the expression of polyglutamines in the protein. Concretely, the protein linked to Huntington disease is Huntingtin, and we are mainly, this is a large protein with very important functions, very diverse functions, but we are focusing on the exon 1, so the N-terminal fragment of this protein that contains the polyglutamine expansion. So this polyglutamine expansion is flanked by the N17, which is a fragment of 17 amino acids preceding polyglutamine, but also this this larger region that is, for obvious reason, called proline-rich region. And both of them have a relevance in the disease, as you will see afterwards. As all the trinucleotide repeat disorders, Huntington disease has a specific features, and the most relevant one is the fact that there is a pathological threshold, meaning that only patients that have more than 35 consecutive glutamines develop the disease. And also the onset and the severity of the disease is correlated, anti-correlated in fact, with the length of the polyglutamine tract. Another interesting feature is what is called the anticipation. Anticipation occurs when the number of glutamines of the offspring is larger, tends to be larger than those of their parents. So we know a lot about how, let's say, how the toxicity and degradation occurs in cells. And I mean, some weeks ago in this webinar series, Hilal Lashwell explained very nicely this work that it kind of illuminated us a lot of on the cytotoxicity. Essentially, what it's telling us is that, I mean, of course, the monomers forms fibrils. Those fibrils form aggregates that grow with time, and those aggregates sequester proteins, lipids, and even organelles like mitochondria and reticulum, endoplasmatic reticulum. After the sequestration, at the end, there's some sort of rearrangement of those inclusion that you can see here when you have an external shell, which is pretty floppy, let's say, that is able to sequester proteins and other organelles, while the inner part is much more solid, let's say. And this has even been imaged in a very nice manner, and you can see it even in 3D. But we are mainly today, we are mainly focused on the previous steps and the steps that go beyond the scheme that I've shown at the top of this slide. And we essentially want to know which is the structure of exon 1, which is the structure of this protein, and if there is any structural origin on this pathological threshold. So when you think in pathological threshold, there are essentially two main models. The first one is called the conformation emergence model. In this model, according to this model, what happens is that given beyond beyond this threshold, in fact, there is a new conformer, a new conformation, new structure that appears, and this is new conformation, which is the toxic one, while the others is not toxic. The second model, trying to relate pathological threshold and toxicity, is what is called the linear lattice model. The linear lattice model assumes that polyglutamine is per se toxic, and the longer is the polyglutamine tract, the more toxic it is. At some point, the homeostatic system of our bodies cannot deal with this toxicity, and the disease is triggered. Interestingly, there are evidences, or let's say evidences for both models using antibodies, but this is a kind of low resolution piece of information. So we are dealing with a low complexity protein, and this has, you will see afterwards, it has many specific features. So if you think in low complexity proteins, those are proteins that have a compositional bias. So the composition is mainly one or few amino acids, okay? And the simplest low complexity protein is a normal repeat, like Huntingtin, in which you have a same amino acid in this kind, the blue one, that is repeated multiple times. But you have many other families. You can have a couple of residues that are repeated, triplets. Those triplets or those repetitions can be consecutive, or they can be even separated by random sequences. And we have up to a tandem repeat, in which you have a long fragment that is repeated multiple times within the protein. Okay, so you can think that this is anecdotal, but in fact, up to 20% of the eukaryotic proteins contain low complexity regions. They are evolutionarily conserved and even promoted. In terms of function, you can think that those proteins have a high concentration of the same chemical feature, and therefore, I mean, this is a way to, the length of this repetition is a way to regulate the concentration of this feature. And normally there is a trade-off between the function that these repetitions allow, let's say, but also the toxicity that they can induce. And the key point is that the sequence structure function relationships for those kind of proteins are, in the vast majority of cases, unknown, because it's difficult to obtain structural information. And this is for the following reason. So, well, the shorter is the, sorry, the shorter is the repetition, the repeated sequence, the higher the probability of disorder. And the more disorder you have, when you have disorder, you cannot use the two classical high resolution methods, which are crystallography and cryo-EM. And then you have to rely on NMR. But even in NMR, those proteins are complex, and you will see why. So just a few words about nuclear magnetic resonance, for those of you that maybe are less familiar to this technique. Essentially, in this technique, what happens is that the nuclei, certain nuclei inside of a magnetic field are able to, the spin levels split, and therefore we can do any, all frequencies, so all the spectroscopic techniques, we can kind of scan and look at the frequency that, that separates the energy barrier or the energy difference between both states. This position of this frequency, which is known as chemical shift, depends on the chemical environment, and therefore contains structural information. If we were only using 1D spectra, this would be pretty useless. The advantage of NMR is that you can use multiple nuclei and you can transfer magnetization from one side to another, and then you can do many 2D or ND experiments. This is the classical one, which is known as the fingerprint of proteins, which is the HSQC that correlates the proton and the nitrogen. And therefore, each one of those peaks corresponds to an NH within our chain. So the first game of nuclear magnetic resonance will be to assign all those peaks, to assign each one of those peaks to the amino acid that corresponds to. What happens when we apply NMR to Huntington? Here you have the NMR spectra, the HSQC, the fingerprint I was mentioning before, for Huntingtin containing 16, 36, 46, and 66 consecutive glutamines. I mean, at the first slide, the first side, you will see that the spectra more or less look the same, despite some differences in resolution. They look the same. However, if we focus in the region in which we have all the glutamines, we will see that for 16, you can more or less identify the peaks, and you could eventually try to assign all of them. However, when you go to a larger number of glutamines, then you start to have broader peaks, and it's impossible to do this individual assignment. So you will see that, so just to mention at this point that in all the samples, or all the samples that I will show to you, all the preparations, our exon 1 is fused to the GFP, and this is to facilitate or promote the solubility of this protein. So we have a question, which is the origin of the pathological threshold that we think is linked to the structure. But at the same time, we have a methodological issue. We cannot answer with traditional methods. We cannot answer these questions because we cannot resolve the structure of our protein, because we cannot assign by NMR all the glutamines of our proteins when they are in the pathological threshold, beyond the pathological threshold. So, and why this happens? This happens because when we produce a protein to do an NMR, we have to feed our bacteria with nitrogen 15. And then you label all the glutamines, and therefore you end up with a situation that I was mentioning before, when there is a lot of peaks, the spectra is severely overlapped. It would be much more intelligent if you could have a situation like that, in which you have multiple samples, in which we individually label each one of the glutamines. In this case, the spectra would be much more simpler, a single peak, and also would have a second advantage, would be that this kind of experiments will be independent of the HOMO repeat length. So this would be a perfect tool, in fact, to study the pathological threshold. I'm going to explain now how we do this kind of labeling. That took us a while to do. So many of you, and probably better than me, you know how a protein is produced, so by cells. So essentially, the sequence of the protein is coded in the mRNA. This mRNA is recognized by the ribosome, and the ribosome is in charge of do the translation. This translation uses these small RNAs, such as a tRNA, that have the anticodon, which is the sequence of nucleotides that recognizes, that reads, let's say, the sequence of codons coded, the sequence of codons of the mRNA. This translation takes place until we encounter, or the ribosome encounters, the stop codon. The main characteristic of the stop codon is that there is no tRNA linked to the sequence, and therefore then there is a protein which is called a release factor that enters in competition, and then the system simply disassembles and the protein is finished. However, if we are able to take out the release factor of our system and we add a synthetic tRNA that has an anticodon that reaches the stop codon, what will happen is that the ribosome will recognize the new tRNA, and therefore the blue ball, the amino acid, will be incorporated into the chain. This approach is called tRNA suppression and was developed by Peter Schultz some decades ago, and is a beautiful tool. And this tRNA has to be orthogonal with the the bacterial system, and normally in this case is taken from this. So practically what we do is the following. So we synthesize we have our synthetic tRNA with the anticodon that reads the stop codon, and we have the glutamine, which is nitrogen 15 and carbon 13 label. We put those two elements in the presence of the yeast glutaminyl tRNA synthetase and the ATP, and in those circumstances, what happens is that the glutamine is loaded into the tRNA. The second element, which is important, is the plasmid that contains our protein of interest, but now at the position that we want to study, we incorporate a stop codon. Those two elements are introduced in a cell-free reaction mixture that contains the transcriptional and translational machinery from E. coli. And the whole process I was mentioning before takes place. And we end up with a situation like this one. By changing the stop codon to different positions, different reactions, different spectra, therefore different assignment, a specific assignment. And this works, to our surprise, the first time we saw it. We see that this is the first glutamine of the Huntingtin containing 16 glutamines. But we did the same thing for this case of non-pathogenic version. We did the same for the 22 glutamines that are inside of this exon 1 with H16, let's say. And also we did the same for a more complex scenario, which is a pathogenic version that contains 46 consecutive glutamines. We can do that not just for the NHs or for the backbone. We can also do that for the complete side chains of C alpha, C beta, C gamma, and the NH of the side chain. And we could assign unambiguously all those atoms for several glutamines, for all glutamines H16 and 16 glutamines of H46. Just a small parenthesis here. So you must think that this is nice, but I mean, this is labor intense and it's a kind of a nightmare to do all those samples. So we kind of also design an approach, but we have recently designed an approach in order to speed up this procedure with a multi-site label. So at some point we discovered that we were able to introduce up to 3 glutamines in our chain. Of course, then you have the three peaks, but you don't know which is which. So our strategy was to produce a second clone in which we overlap one of the glutamines. In this circumstance, what happens is, in this case, Q50, the 2 peaks that overlap between those 2 spectra correspond to Q50, glutamine 50. If we do a third one, but now we overlap 53, what happens is that we can assign unambiguously 53, and 24 we can also assign it because we have already assigned 50. So we know that this is 24. And now, if we keep concatenating experiments, in fact, we can assign 2 glutamines per spectra. In this way, with 9 samples, we can assign up to 18 consecutive glutamines. So we have the chemical shifts, and then we can start doing structural biology. So, sorry. So the simplest way to look at the structure of those systems is looking at the C alpha chemical shifts, and we can tell us, those chemical shifts tell us whether the protein is more helical or an amino acid adopts more helical or extended conformations. When we do that, when we do this for the non-pathogenic version, what we see is that both the N17 and the polyglutamine tend to be helical, have a helical tendency. The maximum of this helical tendency is at glutamine 18, and then systematically decreases up to reaching the polyproline tract. Interestingly, when we look at the pathogenic version, we focus on the polyglutamine. What we see is that the eliciting not just does not decrease, but it reaches some sort of plateau for a long piece, for a long fragment of the polyglutamine tract until reaching the end when approaching to the polyproline tract. NMR is providing local structural information at local level, and we did also a small algorithmic scattering, which reports on the overall size and shape of our system. We did that. We did these size experiments for 16 and 46. And we see, we did some, we observed that this protein is elongated, it's flexible and elongated. Afterwards, we kind of combine this local information from NMR and the global information from SAGS in a procedure to try to derive from some sort of ensemble model that is able to simultaneously describe both data sets. What it came out from this analysis is that Huntingtin in both cases corresponds to an equilibrium between alpha helical conformations of different length. those helical conformations, all of them start around residue 10, and expand towards the polyglutamine tract. Moreover, long helices are more stable, so they are overpopulated, let's say. To have long helices is preferred. The question then came whether polyglutamine is per se helical or there is an influence of the flanking regions into this conformation. So those are the structures. And this is not a question that arises from nothing. It's well known that many biophysical experiments and biochemistry that N17, so the region that precedes polyglutamine, enhances the aggregation of Huntingtin and triggers oligomerization. So if there is any structural connection between N17 and the polyglutamine, that was the question. So we get some clues on this structural connection through the chemical shifts of the side chains. We observe, for instance, that the N epsilon, so the NH of the side chain for glutamine 20 and 21 are clearly shifted, are outside, they are outliers of the general trends of the polyglutamine tract. Moreover, what you expect from a disordered glutamine is that the carbon beta, the two protons in beta, have 2 peaks, let's say, each one peak, while the proton in gamma only present 1 peak. However, when we look at the first 4 glutamines of our tract, those that are connected to the N17, what we observe is a different phenomenon, a different features. For instance, Q21 presents 2 peaks in gamma, and Q20 is the same, while 18 and 19, so the first 2 glutamines, have only display one peak, while you would expect 2 peaks. Very similar scenario occurs with the pathogenic version. So all these features are coherent with a structural model that was developed by Xavier Salvatella in Barcelona, that was a bifurcated hydrogen bonds. So these bifurcated hydrogen bonds are the mechanism by which N17 and polyglutamine are connected, are structurally connected. The main features of these bifurcated hydrogen bonds are the following. So the first one are the canonical hydrogen bonds between residues I and I+4. So in this case, for instance, serine 16 and glutamine 20, or phenylalanine 17 and glutamine 21. So what happens in this bifurcated hydrogen bond is that the side chain of the glutamine is able to fall back and stabilize the hydrogen bond through this second bifurcated hydrogen bond. Okay? And the third element is the presence of this large hydrophobic residue, in this case phenylalanine 17, that stabilizes this bifurcated hydrogen. So this simple model, in fact, explains the vast majority of those observations. For instance, the rigidification of the side chain induces the separation of the frequencies of the two proton gamma residues. Moreover, the presence of the ring currents from phenylalanine 17 induces changes in the chemical shift of those 2 glutamines in the side chain. Another observation came by the fact that it's possible, well, I mean, there are multiple conformations, at least 2 conformations that coexist in solution, and they exchange in a slow regime at the NMR timescale, so in the microsecond, millisecond timescale. We decided to explore a bit more this phenomenon using another trick, which is the use of fluorinated amino acids. So we got this stereospecifically fluorinated glutamine from rhododendrons at the NIH, and we were able to introduce, using the same methodology I explained before, we're able to introduce this fluoroglutamine in position 20 and 21. So fluorine stays where the star is, okay? And then because fluorine has a very different electrostatic electronic properties, let's say, we expected changes in those residues which are in the proximity. So when we introduce fluorine in glutamine 20, what we observe is that the serine 16 moves its position, so indicating that there is some proximity between fluorine and fluorine 16, and this happens for the pathogenic and the non-pathogenic. And when we introduce the fluoroglutamine position 21, what you observe is that phenylalanine 17 displays a second peak. We also did another thing, which is because fluorine is very attractive nuclei for NMR. We did fluorine NMR, and we observed how the chemical shift for Q60 and Q40 is different. We also observe how there are multiple peaks appearing for Q20 and Q21 and in line with the previous observations. And we observe also that Q21 has a very shifted, a very downfield shift. And to explain this thing, what we did was we did some quantum mechanics calculations in which we observed that the measured the downfield, so the most shifted frequencies occur, or expected shifts occur when the proximity of the fluorine, so the distance between the fluorine and the phenylalanine ring gets closer. This is the situation here. So this, in a way, again reinforces the idea that the first glutamine, so glutamine 20 and glutamine 21 are in close proximity of phenylalanine 17 and serine 16. So again, substantiating the idea of this bifurcated hydrogen bond. However, all this data is very qualitative. It's difficult to extract an atomistic description of this phenomena. And for this reason, we decided to do MD simulations. MD simulations of around 20 microseconds MD simulations using this force field that was developed by Jit and Mittal. And this is the fractional helicity that we observe out of these 20 microseconds. And we observe that not quantitatively, but qualitatively, it behaves like our experimental observations. There is a maximum of helicity at N17, and then there is a plateau of helicity along the polyglutamine region. You see how along one of those trajectories, how the Huntingtin goes from an almost random coils scenario to a completely or almost completely alpha helical and melts again. We also observe along this trajectory the presence of bifurcated hydrogen bonds. And those bifurcated hydrogen bonds were more present when the helix was more stable, so it was lasting for more time, indicating probably that bifurcated hydrogen bonds is one of the mechanisms that stabilize helicity. In fact, the presence of bifurcated hydrogen bonds is experimentally observed also in the carbon gamma, proton gamma spectra of many of the glutamines that display a double peak. This double peak, as I mentioned before, is an indication of the rigidification of the side chain and therefore the presence of hydrogen bonds. So experimentally, we observe hydrogen bonds all along the chain, the polyglutamine track, and we observe as well in the trajectory. So we have, up to now, we have been mainly focused on the N17, but what happened on the other side? On the other side, we have the proline-rich region that seems to be important. In fact, it has been also shown that the presence of this proline-rich region decreases the aggregation propensity of Huntingtin. Its absence modifies the fibril structure, and in general is seen as a protective domain. And it's very funny, when you look at the evolution of Huntingtin, what you observe is that for the less evolved organisms, there are almost no glutamines, or just up to 4 glutamines. When there is up to 4 glutamines, there are no prolines around. However, when the number of glutamines increases beyond 5 or 6, then appear the prolines. So it seems that there is a coevolution of those two homomorph repeats, the polyproline and the polyglutamine. So suggesting this idea of the protective domain. So the first question is, so which is the structural role of polyproline? So the first thing we thought was, well, let's try to uncouple both domains. And to uncouple them, what we did essentially was to introduce 5 glycines between both homomorph repeats. If you look at the spectra, there are not many changes. However, when we apply this site-specific labeling and we focus on this glutamine here, which already is partially helical which is a pretty random coil already in the wild type, what happens here is that the shifts move towards directions that indicate that this in the presence of the glycine. So when you uncouple the polyglutamine and the polyproline, this glutamine is much more alpha helical. Okay, so it seems that the polyproline breaks the helicity, the helicity that is present in Huntingtin. If you align the chemical shifts of the two versions, 16 and 46, from the C-terminus, what you will observe is that the chemical shift of both are more or less the same until the point again that there is a more drastic difference. So what it suggests is that, in fact, the taking capacity of the polyproline is independent of the polyglutamine length, which suggests that if you have a very long polyglutamine, you would have a very long alpha helix. Is this true? we designed the following experiment to demonstrate this. So we focus on this glutamine here. This is glutamine blue here. And we produced a Huntingtin containing 66 consecutive glutamines. And now what we did is we label the green glutamine, that is 56, so the same than in the blue one, but also we label the red one, that is at the same distance than the blue one to the polyprotein region, okay? And then the question is whether the blue will be more similar to the green or to the red. And the answer is that the blue one is more similar to the red, indicating that what is really defining the conformational preferences of the glutamines along the track is the distance to the polyprotein track, okay? So, and if you have a very long polyglutamine track, even longer than 66, you would have even a longer alpha helix. Okay, this is structural, biophysics, this is nice, but does it have any relevance in terms of aggregation or in terms of the disease? So for this, what we did is the following. So we designed a couple of mutants in which we kill, in a way, this connection between the N17 and the polyglutamine by introducing two glycines. With those two glycines, we expected to break this helix, to break this connection, and therefore to have a polyglutamine track, which is essentially a random coil. We have another mutant in which we introduce two leucines there. Remember, large hydrophobic residues stabilized bifurcated heterogen bonds. So what we expected is to give more chances to the polyglutamine to do this connection, and therefore to be more helical. Indeed, by NMR, we show that this is true. So the GG mutant is essentially a random coil, while the LL mutant adopts much more stronger helical preferences. Okay. And now we have a system. We have the wild type and two mutants in which the number of glutamines is the same, but the amount of helicity is different. Will they behave different in terms of aggregation? And that's what I'm showing here. So when you take the wild type, the aggregation is relatively slow in the presence of the GFP and have a mid time of 19 hours. But it's clear that when you have the GG mutants on the random coil one, what you have is you slow down the aggregation. While when you have the LL mutant, the tendency to aggregate is much stronger, and it aggregates, in fact, very, very fast. And it's not just a question of kinetics. The fibrils they form, you see that you find after the friction, you see nice fibrils for the wild type and the LL, but the vast majority of aggregates that you find from GG are this kind of heterogeneous things. If you want and look a bit more, you end up finding fibrils. But the vast majority of them are kind of aggregates, heterogeneous aggregates. When you wait up to 5 days, what you see is that those fibrils tend to form these bundles for LL and for the wild type and the LL, these beautiful bundles of fibrils. The GG is also able, but those bundles are less stable, are less ordered, and the fibrils are much smaller. So kinetics and structural fibrils are different depending on the helicity of the polyglutamine tract. What happens in vivo? Well, in vivo, more or less the same. You observe that the number of inclusions found in those cells also depends on the mutant. You see how at the beginning you find those inclusions in the proximity of the nucleus, and at the end they form this large inclusion. Those inclusions can be formed by the mutant and for the wild type and the two mutants. But when you quantify the numbers of cells that present inclusions, what you see is that in all cases, in all time points, the LL mutant have more inclusions, have more infected cells, let's say, than the wild type, and the wild type have more than the GG mutant. So the helix stability not just affects the aggregation kinetics and the fibrils, but also the inclusion, the formation of inclusion bodies and the size of those inclusions. So if we try to summarize in a kind of general model what we have learned, or we have learned in Huntingtin, we see that the key element of Huntingtin is the N17. This N17 is partially helical. This helicity through the bifurcated heterogen bonds propagates the helicity towards the polyglutamine tract, and this polyglutamine tract the helicity is broken by the presence of the polyprotein tract. This is in the non-pathogenic version. What happens in the pathogenic one? Well, essentially the same thing. So the propagation, helix breaking capacity is the same, but the consequence that this helix breaking capacity is the same is that we have longer and more stable helices. And therefore, when it comes to interact with real partners, probably through coiled-coil interactions, we see that we have interaction in the non-pathogenic scenario. In the pathogenic the pathogenic one, probably you also have interactions, although maybe the thermodynamics of this interaction will be different because you have longer helices. But also it can interact with new protein because now the helical tendency is more stable. Also, in terms of oligomers, we can expect that those oligomers are more stable due to the presence of longer helices. And those oligomers eventually form these inclusion bodies that have been nicely imaged, and also, finally, eventually, cause the disease. When we think in terms of the pathological threshold, it's clear that we have shown that the rules governing the structure of the pathogenic and non-pathogenic version of Huntington are the same. Therefore, the linear lattice model is the only one that can explain the situation. So essentially, as a main conclusion, let's say, a bit beyond the pathogenic, the Huntingtin, is that we have developed robust strategies to synthesize proteins with site-specifically labeled glutamines. We have also done with prolines and alines, but I have not explained anything today, by combining tRNA suppression and cell-free strategy. This method is general and only limited to the stability of the protein, and can be extended to quasi-natural amino acids, for instance, the fluoroglutamine, but also fluoroproline we have tried. The flanking regions, N17 and proline region, determine the secondary structure of the polyglutamine. And this, I've not explained this, but these mechanisms are common among the eukaryotic and vertebrate glutamin-rich proteins. And then finally, I think, which is the key point from the biological or biomedical point of view, is that the persistence of the helical region increases with the polyglutamine length and induces and facilitates self-association, both in vitro and in vivo. And here the long list of people that have contributed to this study, going from molecular biology, chemical biology, structural biology, chemistry, SACS, NMR, etc. And this has been a very nice group to work with. And also, importantly, our collaborations at the CBS, from the computational point of view, Juan Cortés and Toulouse, bioinformatics, chemistry, NMR, fluorine NMR, and also quantum chemistry. Thanks all of them for helping us with this issue. And thank you all of you for your attention. I'm waiting for questions. Thank you.
Thank you so much for your great talk. While we are waiting for the questions to come, my question is, for Huntington's disease, I believe there's the disease threshold is 35 or 36 polyQs. You nicely explained with your model how this is being balanced and why, like for small amount of polyQs, you don't have this aggregation. But what is that special about this 35 number? It's all of a sudden just like turns into the starts to become pathogenic. Is it the number of prolines, or what do you think?
I don't know. I think it's a balance. So I guess that it's difficult to answer the question if you think only in Huntington, but you should look at the 9 diseases. And all of 9 diseases, polyglutamine-related diseases, the pathological threshold is different. It's slightly different. I think that is a complex balance between structural issues, like, for instance, capacity of the end-flanking region to induce helicity. This could be one of the phenomena, the polyproline as well, but also probably the concentration of the protein in neurons probably is different among them. So all those parameters, and maybe probably more even, are connected or are probably inside of a complex function that defines this threshold. Having said that, the threshold of 35 is also depends on the age. So probably, maybe there are some people that argue that maybe people with 30 can have the disease, but maybe they would develop the disease when they are 120 years old. So probably the toxicity is accumulative, and now we observe is 35 because we live around 80 years old, but maybe eventually in the future, maybe this threshold will go down, let's say. So I don't have an answer, a clear answer for your question, but I'm sure that there are many parameters that play a role. Sorry.
You start having questions. The first one is for anonymous attendee. Do you think the lattice model is also relevant for other repeat disorders or specific to Huntington?
I think that is general. But it's just a feeling. So so you know the difficulty is so is to study the pathogenic version. So this is the first, I mean, and the only way to do it properly at the residue level is probably the approach that we have followed. And this has already been done for Huntingtin. But in the same way that the mechanisms that induce structure in Huntingtin, I'm sure that they are also applied, are known to be applied, for instance, to androgen receptor, most probably will be also applied to the other 7. I'm almost convinced that the lattice model, so the structural rules will be the same for all the diseases, but it is true that this is just a speculation.
We have a question from Christian. He says, hi, Pau, very beautiful study. Given the surprising dispersion of the cues, would you have had the possibility to work with full labeling, or is the selective labeling necessary? And there is a second sub-question. Do you want to read it, or you would rather answer this one first?
I will ask the first one. Thanks, Christian, for the question. So probably for a limited number. So let's put it this way. So for a limited number of glutamines, maybe you could do the same for using a complete labeling. And in fact, Lucio Friedman did something like that with an exon 1 with 17 consecutive glutamines, with using a trick of the pH dispersion, so increasing the dispersion of the spectra using pH change. This is possible for 17, but I'm convinced that you cannot go much farther than that. Having said that, for androgen receptor, which is much more structured, so it's much more helical than Huntingtin, Xavi Salvatella was able to assign, using carbon detected experiments, he was able to assign an androgen receptor fragment containing up to 25 glutamines. He couldn't go beyond this number. So I think that if you really want to address pathological threshold, then yes, you need an approach similar to the ones we have done. And the second is, it seems that sorry.
Yeah, do you mind if I read it? Did you compare it with a poly-Q chain without anything flanking? I would expect then that everything overlaps different from what you observed for the more native poly-Qs.
Yeah, so when you don't have flanking, when you don't have the N17 flanking, this is almost equivalent to our GG mutant. So in our GG mutant, this connection, a structural connection between the flanking region and the polyglutamine is not possible. And then what you see is that all the broad banana of the polyglutamine just kind of collapses in a more in a peak that is less helical. So I would expect that this behavior would be equivalent to the one if you only have polyglutamine alone. I think that I have not compared exactly the chemistry. Annalisa Pastore has done of different lengths alone, and they behave more or less like this. So if the hidden question is whether polyglutamine is per se helical, the answer is maybe a bit, but not that much.
And there is, the third question, and this is the last before we move to the second speaker. For Christian, it's kind of an interesting question. It seems that proline extension prevents aggregation. Why did nature not just extend poly-P at the appropriate rate as poly-Q is extended to avoid complications of aggregation?
Yeah, that's an excellent question. I think that nature did its best, let's say, which is introducing multiple glutamine, multiple problems after a glutamine track to try to minimize. So I guess it's a fight, no? So I guess in nature, on one hand, you have seen in the evolution that wants long polyglutamines because this probably induces elicity and allows new functions for this protein, probably through coil. But at the same time, detects some potential problems and puts probably problems. And it works for the vast majority of us. What happens is that through a phenomenon that is not linked to this, which is the DNA slippage, sometimes the polymerase gets messed and introduces some glutamines more into the system. So, and these at the end, I mean, after a few generations, this can cause the disease. So it's a good point. I think that the nature already has done this thing, but maybe to an extent in which a certain population get to up to some point risk to develop this disease. I don't know if I answer your question, but
yeah. Thank you very much for the great talk. And right now we are ready to move to our second speaker, Dr. Vijay Rangachari. And while he's uploading his presentation, I'll just start my introduction. Dr. Rangachari is a professor of biophysical chemistry and a director of the Center for Molecular and Cellular Biosciences at the University of Southern Mississippi. Dr. Rangachari did his PhD in biophysical chemistry at the All India Institute of Medical Sciences, New Delhi, India. After spending 1 year as a visiting scientist at the Institute of Molecular Biology, Friedrich Schiller University, Jena, Germany, Dr. Rangachari moved to the Institute of Molecular Biophysics, Florida State University, for his postdoctoral studies. His current research interests are biophysical and biochemical studies of intrinsically disordered proteins, amyloids, and liquid-liquid phase separation. Thank you very much for joining us today, and it's your turn, and nice meeting you.
Thank you, Magda. Can you hear me well?
Yes, it's perfect.
Okay,
Everything is perfect. Your slides look nice.
Awesome. Great, thank you.
You're ready to start.
Well, first of all, I want to thank you for the great introduction again, and the invitation to present my work here today. And specifically, I want to thank all the organizers, Rams and you, for this extraordinary series of seminars that you've put together for the last 2 years. We've been enjoying week in and week out. It was really a great service. Thank you very much for that. Today I want to talk about some of the work that we've been doing quite recently, actually started between, that is to look at the interactions between alpha-synuclein and TDP43. As you will see, there's a lot of open-ended questions at the end of this talk, and I'll appreciate if you have any good feedback from you guys. But I think I hope to convince you that I think this heterotypic amyloid formation is pretty important in the field of amyloids, and moving forward, in some cases, perhaps even more important than the homotypic ones. So, okay. Well, I'll start with this more familiar slide for this audience, is that we all know that the specific hallmark of neurodegenerative diseases mainly involve the deposition of amyloid proteins, and a specific amyloid proteins which are in majority in the lesions that are formed in the brains and that are categorized as such. And I think each of the neurodegenerative diseases listed here have a specific protein that is a deposit as a amyloid plaque in the brain. For example, in Alzheimer's disease, you have this amyloid beta. In Parkinson's disease and Lewy body diseases, we have alpha-synuclein as a majority of the protein that is involved in amyloids. But in reality, as we have come to this in the recent past, what we commonly observe is not just one protein deposit, but there are multiple proteins involved in some similar amyloid formation, sort of concomitantly with the others, right? This example here that I'm showing from the review from COVAX, we showed that the 93-year-old patient had the deposits of amyloid beta, phosphorylated tau, alpha-synuclein, and TDP43, sometimes co-localized and many times in distinct regions of the brain. Now, I think in the same review, it's remarkably summarized table is that when you look at these list of 18 or so neurodegenerative diseases, what you can appreciate here is that the blue bar represents the percentage of a single majority protein or a homotypic amyloid formation, whereas the yellow bar represents the combination of more than one protein involved in amyloid formation, right? So you can see that there is a significant increase as you go down this list, the presence of more amyloid protein, more than one amyloid involved in the pathology. And again, as you can see in the top, the bar that represents these color-coded proteins, and you will see there's majority of these co-localized amyloid deposits involve more or less these combinations of these proteins, tau, alpha-synuclein, TDP43, and sometimes PRP. So the importance of that can also be visualized by looking at this pie chart. And if you look at the tau and A beta that's been known for a long time to be interacting with one another, and mainly in AD, but if you look at the tauopathies and alpha-synucleinopathies, you have a significant overlap in many pathologies, including CBD, PSP, and more lately, there is something called late. I'm going to talk about this shortly, multiple system atrophy and so on. And you have a significant overlap between the two deposits of tau and alpha-synuclein. And more recently, and this list has actually included this TDP43, the emergence of the observation of having TDP43 deposits in these diseases have significantly increased, and more is actually emerging. And the interesting aspect is that there are not just simple co-observation of deposits of these proteins, and most commonly you will see a co-localization of these amyloid deposits as well. And some of these have been documented recently, and as this couple of slides, a couple of papers that I'm pointing out here. More importantly, I think the paper from Yokota, who actually looked at this amygdala regions of the brain of LBD patients, show these distinct co-localization of cytoplasmic co-localization of both TDP43 and alpha-synuclein. And of course, you will see, as pointed out here in the small arrow, you can see these distinctly different homotypic inclusions which of the two proteins of alpha-synuclein and TDP43, but you'll also see a significant overlap and co-localization of these deposits. And that is an important aspect, and whether this co-localization is sort of a serendipitous observation, or do they have anything to do with molecular interactions between the two. More importantly, the recent paper from the late Trojanovsky showed that the limbic predominant age-related TDP43 encephalopathy, also called late, has a significant overlap in terms of the TDP43 and alpha-synuclein proteins. And in specifically, they point out that some of these neurites are co-localized with phosphorylated alpha-synuclein as well. So now these the reports that I've shown you is convincing enough to look at the possibility of a molecular interaction between the two proteins. But if you actually take a step back and look at the more biophysical aspect of protein aggregation, we all know that the aggregation of proteins have their own sort of a aggregation funnel, which is kind of in the red area of this picture. We know that these proteins actually can be modulated in multiple different pathways that can end up in different polymorphic structures of amyloids. And this is not new, and it's been well documented over the years. And we also know that there are several different modulators for this process. You can use the small molecules to all the way to large proteins to modulate this sort of energy landscape to end up in polymorphic structures, one kind or the other. And if you include in this modulator list another amyloidogenic protein, which makes very intriguing possibilities because both are, when both amyloidogenic proteins are pathogenic, are involved in neurodegenerative diseases, and you can end up having a significantly modified landscape, and specifically with a particular type of polymorphic strains, which are not homotypic, that are sort of different, that are polymorphic in their differences in their structures, but rather having some level of co-localized, co-interactable polymorphic strains. In theory, it's possible, and that's what intrigued us to look at this idea behind the possibility of TDP43 and alpha-synuclein interactions. Again, I'm showing you some of the structures just for to capture the different types of fibrils that one can end up having. And these are the list of 3 that I just picked up, and there are more. And again, one from the Eisenberg's group and Solberg's group and so on. And all 3 happen to be the recombinantly generated fibrils from recombinant proteins of alpha-synuclein. As you can see, there are significant differences in them, depending on the method in which you grow these fibrils. You also have predominantly, very importantly, 2 different structural polymorphs that have been known so far about TDP43 C-terminal fragment. I'll come back to that C-terminal fragment a little bit later. But if you look at these TDP43 fibrils, again, on the left, you will see the one that is generated from the brain, the recombinant protein with the seeds derived from the patients. On the right, you will see a purely recombinantly generated structures of TDP43 fibrils. And of course, you can see the significant difference in them. So such polymorphs are important, and they have a clinical ramifications as well. But I think the important question is that why, how TDP43 could interact with alpha-synuclein. So to do that, let's look at this biogenesis of TDP43. This is a protein that is classical ribonucleoprotein present in the nucleus. It's got an N-terminal domain, 2 RNA recognition domain, and a C-terminal low complexity domain, or prion-like domain that actually involved in aggregation. But in the nucleus, it is involved in a variety of different functions, including transcriptional regulation, mRNA splicing, and biogenesis. In fact, microRNA biogenesis is also something that this protein is involved in. And in pathology, the protein is translocated to cytoplasm, and where it undergoes specific enzymatic cleavage to release these C-terminal fragments, broadly defined as C25 and C35. There are many more fragments than these, and these end up forming insoluble inclusions that are cytotoxic. The other side of the story about TDP43 is that the TDP43 is also translocated to cytosol under stressful conditions by coacervating with the RNA molecules along with the many other proteins, and end up being in a phase-separated membraneless organelle called stress granules. And these stress granules involve not just TDP43, there are many other proteins in there, alongside a concentrated foci of RNA molecules. So the question now remains is that whether these membraneless organelles and the inclusions have any interactions between them, any synergy between these two. Can TDP43 convert to insoluble inclusions from stress granules, because you have created a concentration gradient by means of packed in the dense foci of a droplet, but could that promote aggregation is something that is not known clearly. And also not known is whether these fragments are involved in stress granule formation in phase separated in manner. So that is also not clear. So we are interested in these questions. And of course, if you look at the alpha-synuclein biogenesis, we all know that this alpha-synuclein forms this cellular intra-cytoplasmic inclusions called Lewy bodies. And these also, the protein is also present outside the cell and also in the membrane-bound form. So alpha-synuclein is somewhat ubiquitous in terms of its locations in the cell. So of course, the interesting part is that, of course, we know that the Lewy bodies are in the cytoplasm, and we also know that TDP43 forms inclusions in the cytoplasm. So can they interact with one another is the question that we're interested in. And so we set out to ask this question and answer this question, rather. Do alpha-synuclein and TDP43 influence one another in amyloid formation? So to do so, we started looking at this from a very simplistic manner. So if you look at this simple homotypic aggregation model that is depicted like this, and you can also conceive a model where two proteins are equally interactable, and then you can end up having at some level of interactions between the two proteins, and that two get incorporated in the evolving amyloid fibril, right? So the question is, okay, can we probe this mechanism? So we looked at three areas or the regions of these interactions. So one, in the monomeric level, can the monomers interact with the two, and how they can actually evolve into a structure like this in a heterotypic interactions? And can the monomer interact with some intermediate? In this case, we talked about oligomers. Can the oligomers interact with monomers? And also fibrils, can the fibrils interact with monomers? So we want to dissect these in three broad areas of this aggregation pathways. Of course, we wanted to use, for this study, we looked at use this full-length alpha-synuclein, and we've also used predominantly the C-terminal fragment of prion-like domain of TDP43 because that was the one that is involved in inclusion formation. So we first looked at the monomer interactions by looking at the HSQC spectrums. We, in this case, we labeled the uniformly labeled alpha-synuclein with N15 and added the unlabeled TDP43 and compared the spectra of the two and saw there are significant differences when you actually add the TDP43 in equimolar stoichiometric equivalence. And as you will see, there are two things we noticed. One, there were disappearance of peaks, and two, there were significant shifts in some of those chemical shifts. And so we thought that this is probably because of clear one, that is they're interacting, and they're probably are promoting could be promoting aggregates. And we looked at the other way around. We labeled TDP43 with the uniformly labeled N15 and added unlabeled synuclein in this case. And again, you saw the significant disappearance of the chemical shifts here, or peaks, which sort of indicated to us that it's a possibility of the size increase because of the disappearance of these peaks. So now when we want to actually confirm this, we looked at these simplistically look at the dynamic light scattering data, and you see that the monomer by itself, alpha-synuclein and the TDP43 have small sizes, but as soon as you add the two in equimolar the stoichiometric proportions, and you end up having a significantly large species. And it told us that it is clear that these are interacting, not only interacting, they're actually promoting aggregates of some kind. So we carefully dissected by looking at the titrations of one protein over on the other, and look at and by changing the stoichiometries, and saw that in this case, what you see here, the co-incubated samples are indicated in the filled black circles, and you can see as compared to the controls, the co-incubated samples have decreased lag times, indicating that they are promoting aggregates much faster than the homotypic ones. And that is in the top panel is when we actually kept alpha-synuclein concentration constant and titrated TDP43. On the bottom, you see the reverse where we kept the TDP43 concentrations constant and then titrated alpha-synuclein. So in both cases, we saw that the co-incubated samples seem to have much more faster aggregation than the homotypic ones. And then just to look at this a little more carefully, we took the alpha-synuclein sample because alpha-synuclein's lag times are homotypic lag times are quite large and long, so that's why they don't aggregate fast. So we added substoichiometric amounts of PRLD monomers to this, and we clearly identified a decrease in the lag times, and you can see that this is the promotion of aggregates by substoichiometric amounts of PRLD. And that can be, of course, we can look at these microscopic images and stain them with THT, in this case, thioflavin S, and we saw that the deposits are clearly amyloid type. And we wanted to quantify this by quantify these deposits. So we sedimented these deposits and actually did the mass spec. In this case, this was MALDI we used, and looked at the relative intensities of these peaks. And what we saw was when we sediment these deposits and looked at the intensity of the incubated samples were much greater for alpha-synuclein compared to the control, suggesting that the alpha-synuclein is actually pulled down along with the fibrils that are formed. And the same way, we also did this for the TDP43, and we saw this exact same kind of increase compared to the controls. When you add this TDP43, they are also getting pulled down into the deposits. So we know that they are getting deposited in insoluble inclusions. The other thing that we saw was that there were, if you look at these, we started calling these hybrid fibrils because at that time we were kind of bold to claim that way, but I think we wanted to see that when you look at 1:1 stoichiometric amounts of monomers, we ended up having equal intensities of these proteins in these sedimented fibers. Well, of course, that doesn't tell you that these are hybrid fibrils, but we saw that the first indication of that, and then we wanted to confirm this, whether they actually are incorporated within the same fibril. So to do that, we actually used this gold immunolabeling technique, which is fairly well established by Hilal Lashwell's group. That so you take this. In this case, we took the TDP43 and alpha-synuclein, which is histidine tagged, and a gold nanoparticle can be attached to this antibody that recognizes the histidine tag. By doing that, we can actually specifically form the fibril and then add the gold nanoparticle to see whether you can actually identify the presence of synuclein and PRLD in the same fiber. So we did that. On the left, you see this control PRLD on PRLD, which is homotypic ones, and you saw that that works. On the right, in this case, the panel J, what you see here is the combination of both PRLD and alpha-synuclein, and you can see the single fibril stretch containing alpha-synuclein gold nanoparticle. That suggested to us that we have a somewhat what we call this hybrid fibrils. We still don't know molecularly they form, what kind of structure they form, but we do know that they are actually forming some kind of intertwined, having both proteins present in the same fiber. Now, with that, I think we then sought to look at the interactions and the intermediates. In this case, we wanted to see the interactions of the monomer with oligomers. And unfortunately, we could not isolate or identify even oligomers of TDP43 PRLD because it aggregates really fast, it sticks to the column and things like that. We could not isolate it. We cannot even see an oligomer, at least in the conditions in which we generated these samples. But on the other hand, alpha-synuclein oligomer preparation has been quite well documented, well established. And for this, we actually used one of these metabolite called DOPAC, and which is also known to interact and known to form alpha-synuclein oligomers. And here I'm showing that some data that we got, indeed they form nice oligomers. They have a series of bands across the 40 kilodalton to about 110 kilodalton. And if you look at the atomic force microscopy images, you can see that the absence of any long fibrils, but you can see these punctate dots of oligomers. And we use these oligomers to seed TDP43 monomer. In this case, we used 15 micromolar sample here shown in blue. When you add oligomers, you can immediately see that there's an increase in THD fluorescence, indicating that they are seeding the aggregation of PRLD. And of course, that can be captured by Western blots, and you can see that the fibril formation is very clear. The unspun sample have this high molecular weight bands on the top of the gel compared to the control. And we can also look at this transition from a random coil to a beta sheet structure through FTIR, in this case, going from 1643 to 1615 as we seed the alpha-synuclein oligomer. So it's fairly clear to us that the oligomeric sample of alpha-synuclein is also able to interact with alpha-synuclein, at least one type of oligomer. Of course, it doesn't really encompass all types of oligomers of alpha-synuclein. So the last part was, okay, what about fibrils? So we generated fibrils and isolated them, sonicated them, washed them, cleaned them, and then we have the sonicated fibrils to use, and then used as a seed to see the kinetics of aggregation. What I'm showing you here is the addition of the TDP43 monomers on preformed fibrils of alpha-synuclein. And in the inset, you'll see the homotypic ones. There's a homotypic control. So you can see that in the concentration dose-dependent manner, if you increase the concentration of the seed, you can increase the kinetics of seeding aggregation. But if you look at the filled circles in the graph, those are the heterotypic ones. And of course, as we expected, the PRLD did not actually increase the rate of aggregation immediately, as one would expect for some kind of elongation process, but rather we saw a clear lag time, and then there is a significant increase in THD, so which tells us that some sort of a cooperative mechanism is happening between the heterotypic seed and the monomers here. But nevertheless, we saw some seeding. And if you do the reverse experiment where you use TDP43 fibrils to seed with the alpha-synuclein monomers, so of course the open circles that you're seeing are homotypic controls, so you can see that increase in the rates. But if you look at the heterotypic ones at the bottom, we never saw an increase in the rates of aggregation or seeding at all for the reverse titration. So that was surprising to us at the beginning. In fact, we've collected data well beyond 10 days, and we don't see any increase in the aggregation. So it doesn't really seed the PRLD fibrils do not seed alpha-synuclein monomer. So that was kind of surprising to us. And if you look at the same reactions in FTIR, it also validates what we see with THD fluorescence. As you see that 1650 and 1643, the alpha-synuclein monomer and PRLD fibrils do not change the conformation, whereas when you look at the other titration, alpha-synuclein fibril added PRLD monomers converted immediately or with time to a 1615, showing a parallel beta sheet formation. And of course, this can also be captured with atomic force microscopy, and you see the same reactions here. The alpha-synuclein fibrils on the left here. When you seed that to PRLD monomers, you see these well-formed, well-defined fibrils. Whereas the other way around, when you use the PRLD fibrils to seed monomers of alpha-synuclein, we do not see any mature fibril emerging, and which kind of gave us an idea that this process is somewhat selective in terms of seeding. So now the question now is that, okay, well, when we know that they are interacting, they're seeming to form some heterotypic aggregation, whether that is kind of modulated by the stoichiometry and the kind of aggregates that we add. So we wanted to actually go deeper into this and ask, well, so what kind of polymorphs are generated, if any, right? So we looked at the biophysical differences between these. So we used what we call these hybrid fibrils made by stoichiometric equivalent monomers to begin with. And then we also had a alpha-synuclein seeded PRLD fibrils, which we know that they seed very well. And then as controls, we use the homotypic alpha-synuclein fibrils and TDP43 fibrils. So when you use these samples, we saw the differences first with the different dyes. This is intrinsic tryptophan that showed significant differences in their intensity and a slight blue shifted depending on the sample that you use. The green one is the hybrid fibril, with the red one is the alpha-synuclein seeded PRLD fibrils, where other ones are controls. And you can see the same type of differences, not exactly identical differences, but we do see tangible differences of intensity and shifts with ANS, curcumin, or the dye DCVJ. And we also looked at the stability of these fibrils formed by limited proteolysis by using proteinase K digestion. And this is a Western blot of those reactions. And you see this, the alpha-synuclein PRLD are the controls. And on the right, you see this one-to-one hybrid fibrils and seeded PRLD fibrils. You see the banding patterns significantly differ. And to look at the lower molecular weight range, we resorted to MALDI analysis. And you see the top two panels are the controls of alpha-synuclein and PRLD. And the bottom two panels are the seeded samples. And what you see here is the red stars represent the absence or the disappearance of the masses that we saw with PRLD control. So they're no longer there in the hybrid fibrils. And the green arrows indicate the new peaks that are forming. So what we can take from these results is that we can say that the banding or the proteolytic patterns are different. Is possibly because the structures of these aggregates are possibly different to accessible to proteinase K enzyme. So one other thing that we did was to take these fibrils and analyze their stabilities sort of in an indirect way. The way that we did this experiment was to take the beta sheet forming fibrils and then use SDS to actually denature it. So use the detergent as a denaturant, because we know that adding SDS can actually convert these aggregates to alpha helical structure or a 310 helical structure, so depending on what type of aggregates they form. But that transition of beta sheet to helix can be monitored as a function of temperature. So we get the relative stabilities of these fibrils. So that's what I'm showing you here in CD. You can see from this black curve is the actually initial beta sheet structure that when you add SDS and melt, so to speak, by increasing the temperature, it forms a helical structure as a function of temperature. So we did that for all of these controls and the fibrils, and what we saw is summarized on the right. So the way that we represented this was the best to actually look at the differences in ellipticity between the 208 and 218, which is actually the differences in the beta sheet extent and the alpha helical content, or a helical content, I should say. It's not exactly alpha helical. But you see that the black line represents these homotypic PRLD fibrils, and you see this, the temperature of melting is somewhere in the range of 60 degrees. When you look at the PRLD monomers, which is in green, it immediately melts. So more towards the bottom, the lower value represents more helical content, the higher value represents more beta sheet content. And you can see that they immediately melted. But the others, the hybrid fibrils and alpha-synuclein seeded fibrils, both have relatively less stable than the homotypic counterpart, but they have differences in their stabilities where the alpha-synuclein seeded fibrils are slightly more stable than the hybrid fibrils. Right? And to take home message here is that we do see that when you do a seeding of alpha-synuclein or PRLD in different stoichiometric amounts, or the nature of the aggregates that you're adding, it differs the eventual fibril that is emerging, at least in terms of biophysical property. So to be sure that we do see some differences in the fibril structures, This is work done by Dr. Anand Paravastu and his graduate student at Georgia Tech. It's a wonderful collaboration that's ongoing. And so he used solid state NMR to see the differences. In this case, we did this dipolar assisted rotational resonance and dipolar recoupling between C13, C13. So here we are just simply labeling the TDP43 with C13 uniform labels and added the unlabeled alpha-synuclein sample. So on panel A, you see the homotypic control of PRLD fibrils. And of course, you see these cross peaks, which are typically generated based on the C13 dipolar coupling, which are closely spaced, maybe within the same residue or the neighboring amino acid residues. So in the amyloid core, probably that's what you see in these circled areas. But you see the same circled areas with the hybrid fibrils and the alpha-synuclein seeded fibrils. You see significant differences or disappearances of these cross peaks, indicating that their amyloid core differs, at least in terms of the structure. So of course, this is not a really high resolution or a structure that we have done, but we got enough data to sort of support our previous conclusion that you do have a different type of polymorphic structure that is emerging by modulating the types of alpha-synuclein seed that you add. So, well, these are all good. Now, the question is what happens in cells, right? So do they co-localize in cytoplasm? That's the first question we asked. And this is a very simple experiment where we actually transiently expressed TDP43, at least the PRLD C-terminal fragment in neuroblastoma cells. And then after the transient expression, we pulsed these cells with the alpha-synuclein monomers and then looked at the co-localization in there. So both were labeled orthogonally. And in this case, you get blue fluorescent protein labeled PRLD. You can see these, the formation of a significant number of insoluble inclusions in the cytoplasm. And this is, of course, the nuclear stain. And so if you add alpha-synuclein, which is labeled in yellow, you can see that there is a fairly a good co-localization of these two proteins. Of course, there are deposits of alpha-synuclein which are outside this PRLD puncta as well, and that is quantified here. So we know that they are co-localizing. Then what about the toxicity? And we did this very general or minimal cellular assay. As we all know, the MTT assay is not something of a foolproof, but it does give some indicators of whether this is viably or is it more or less toxic to the cells. So here, what I'm showing here is that the superscript M means fibrils, O means oligomers, F means fibrils. So you can see that the different stoichiometric proportions of the monomeric control, heterotypic monomers, and alongside this controls, and you can see the interesting aspect is that only those heterotypic samples, which is I'm showing you in the boxed areas, in those samples, you can see that the heterotypic samples have are more cytotoxic than the homotypic counterparts. The only exception was the oligomer sample, and I think we believe it's because oligomers are highly toxic in either case, and we didn't find any statistical significance between the two. But all the others, in case of fibril seeded samples and the monomers, we see the heterotypic ones have more enhanced and pronounced toxicity to the cells. And finally, this is a work done at Rakesh Kayad lab in UTMB. So Rakesh's lab used these primary neuronal cultures to look at the synaptic markers and see whether the heterotypic ones have any specific interactions or disturbance of these synaptic connections. So he used two different types. One is the presynaptic marker synapsin 1 and the postsynaptic marker PSD95. And what the results are summarized at the bottom. Specifically, what we saw was the PSD95 significantly lowered, the postsynaptic markers are significantly lowered in terms of the alpha-synuclein fibril-seeded heterotypic fibrils compared to the homotypic ones. And of course, the co-localization of this PDP75 and synuclein also showed significant difference. this suggests that so this is a selectivity of heterotypic aggregates to cause certain synaptic dysfunction is seem to be significant. So this in general, I think, convinces that there is a selectivity of these heterotypic aggregates to do the cellular dysfunction as well. So the last part was we asked the question whether the alpha-synuclein modulates the condensates, which are a part of the stress granule. So to do so, we looked at the formation of liquid droplets of this TDP43 by adding RNA. In this case, we added poly-A RNA, and we saw these clear droplets that are formed with the colocalization of the two. And using that as models, we sort of titrated alpha-synuclein to this in two different proportions of stoichiometry. One is 20:1 and 20:5 substoichiometric proportions. And you can see that these droplets are formed, and as soon as you add alpha-synuclein, immediately they distort and deform these droplets to certain levels. And you can see that alpha-synuclein seeming to be colocalized with these droplets as well. And if you let this evolve for 24 hours, the changes are even more dramatic, where the droplets of the control seeming to be holding good. But when you see the addition of alpha-synuclein after 24 hours, you can see the significantly deformed droplets more look like somewhat insoluble deposits or even gels. To quantify that, we looked at the FRAP data, fluorescence recovery after fluorobleaching. So when you do that, you see the kinetics of the dynamics inside the droplets have dramatically changed. So in the top panel, in the control, going from 0 to 24 hours, you do see some truncation of the dynamics, which suggests that there's some sort of a gelation of these droplets over time. But when you add alpha-synuclein in these substoichiometric proportions, you completely negate any of those FRAP recoveries, suggesting that you're immediately forming some sort of gels or even aggregates. And to see whether their aggregates are amyloidogenic, so we looked at the THT profiles of these reactions, and we saw that the THT comes up, suggesting that these aggregates seem to form amyloid-like structures when they actually become solidified. So at least it's minimally, it's nothing related to an in vivo situation, but at least in vitro, we can claim that there is a propensity for alpha-synuclein to interact with these droplets, RNA TDP43 droplets, to enhance amyloid formation. So with that, I just want to conclude with this story. Again, as I said, it's a fairly recently started project, so we don't have a deeper understanding yet. But I think what I hope I've convinced you that this phenomenon is real, and we can see that there is a selectivity going on. Alpha-synuclein monomers can interact with PRLD monomers to form this sort of heterotypic aggregates. In this case, with the monomer case, I think we call it the hybrid fibrils. We know that all oligomer of alpha-synuclein can also interact with TDP43 monomers to form some kind of an aggregate, but these oligomers are not sort of bona fide, but they're induced by DOPAC, so we're not very sure how relevant it is for in pathology. But we do know that the sonicated fibrils, and the fibrils are not are able to see TDP43 monomers, but the reverse is not true. So the fibrils of TDP43 are unable to see alpha-synuclein. And it's kind of also interesting that the condensates between TDP43 and RNA can also be modulated by alpha-synuclein. Whether they actually go into this droplets or not, it's not very clear. We know that they're colocalized. We are looking into these things at this point, but we know that they can actually make these droplets somewhat into amyloid fibrils by interacting with that. Just to give you the same, this is like a data-less conclusion, so to speak. We don't have any data for this. These are actually the PDBs of these already shown polymorphs of alpha-synuclein. And I think it is fair to think there's a possibility of conformational selectivity in terms of seeding as well. We're not sure whether all of these alpha-synuclein aggregates can seed. There is a selectivity process in the compatibility of the structures to form a template with PRLDs, also probably playing a role in terms of what they can actually form and what kind of structure that it evolves eventually. So we don't know the detailed structures of these yet, and these are all ongoing investigations, and hopefully we'll get to know the answer sometimes. So I will just conclude that with the work that I just showed was published in 2 papers. I think, as I said, it's a recently started work, so we got only 2 papers on this. If you're interested, and please take a look at these. Let me know. And I want to thank the people who actually did the work. This work was spearheaded by my graduate student Shailendra Dakhal. He's about to graduate next year, and Mala is just starting to work on that project again. And so there's some talented undergraduate students that are working on this project also. And this work cannot be completed or could not have been evolved the way it is evolved without the collaborative effort of Anant and Rakesh. Both have been phenomenal in having this project going forward. And I want to thank the funding agencies for the support, and I'll be happy to take any questions. Thank you.
Thank you, Vijay, for the great talk. I really enjoyed it. While we are waiting for the Q&As, I have a question. I might have missed it. Did you use the 25 kilodalton or the 35 kilodalton of TDP43?
Oh, we used actually the 18 kilodalton. I'm sorry, I did not.
Oh, you did the,
mention...
you used the 18 kilodalton because I was thinking whether, depending on the size of the fragments, this polymorphism will change.
Yes, I absolutely agree with you, because the 35 and 25s are extremely difficult to work with. I mean, that's one of the reasons why we shied away from, because they're quite insoluble and they're not stable in many different buffers. But having said that, we have some success with 35, but definitely not 25. But regardless, I think the PRLD, we want to first find out what the role of this low complexity domain. So I think the structures of that is available, so we can correlate with that and things like that. So that's the premise at which we started. We wanted to do work primarily with the prion-like domain at this point. I think we need to go into these other constructs as well.
And do you have any speculation on which region of alpha-synuclein will be responsible for the seeding? I'll put it this way: again, you might have mentioned it. Which polymorph of synuclein have you tried different polymorphs, for example, to see whether you will get the same effect?
Yeah, no, I think that's a great question. I think we have an idea. I think solution NMR, when you do the monomer interactions, we know that from that, we know what regions are actually interacting and what regions are actually promoting aggregation. The amyloidogenic region in the middle, actually, you don't see the disappearance of peaks are mainly there. So assuming that there is some interaction there, but the peripheral interaction where the other regions of the alpha-synucleins are also interacting for sure, but they are not probably in the core. I think that's what our guess is. So we still have not gotten into that. But again, the actual data comes from the solid state NMR. We know what residues are probably involved in PRLD, and we haven't gotten to the alpha-synuclein side of it yet.
Okay, yeah, thank you. Let's see. Anybody from the panel who has a question? Danilo, Carmelo, Sam, do you have any questions for the speakers?
Yeah, really a massive number of data. Probably I missed something. But I wonder, beside the notion of comorbidities in proteinopathies, I wonder whether there is a common ancestor really before protein aggregation, something when you lose the control of protein levels. Because when you have an excess of protein and there is an hydrophobic environment, everything collapses. So I would like to know your opinion on this rationale.
I think it's a very realistic possibility that there is something up the hierarchy of things, so to speak. But I also believe that the basal level of these aggregates have a lot to do with the interactions, because I think it is when we do the in vitro experiments, obviously there are very biased in terms of concentration regimes. But if you look at the basal levels of those expression levels of TDP43, and that is enough for seeding interactions. I think that is the key part, I believe, is, when I showed you the data of the 1:1 stoichiometric equivalence is probably the farthest from pathological conditions. But I think if you look at the seeding conditions, you can actually see that even the substoichiometric levels of one protein over the other is able to seed. So I do believe that this, although there can be an upstream event that is actually causing these, the levels of expression of the aggregation prone proteins have some significant interactions among themselves, will have an effect on what the polymorph is generated. I think that is my best guess. But yeah, I agree with you. I think there are so many upstream events, so to speak, or the clearance events that are probably involved in such polymorphic strains and interactions as well.
Okay, thank you.
I see that you have a question on the Q&A.
Just
Carmelo.
curiosity, yes.
You can go ahead. Yeah.
Nice talk. Thank you.
Thank you.
Have just one question. In your opinion or in your experience, you have taken into account the role of lipids on fibril formation, because was reported the structure of alpha-synuclein fibril trapped some lipids.
Mm-hmm.
Have you explored this field, or what is your opinion about this?
No, I think it's a great, interesting point. I think lipids, I think it's a dimension that we have not included. In fact, there are several dimensions we have not included. One, the post-translational modifications is something that we are looking at right now. That's one dimension of it. The second one is the lipid aspect of it, because we know that there is association of lipid with alpha-synuclein clearly. And in fact, TDP43 also is known to form lipid droplets. So now the question is, what is the role of lipids is a very pertinent and valid question. So we haven't gone into that, and we're very interested in it. Of course, there are multiple directions this can actually, we have to investigate. And I think we are not there yet. I think but that's a very realistic possibility. And I believe that lipid interaction is super critical. In fact, that's another parameter that can actually change the structure that is eventually formed, which may be even more relevant in pathology.
Okay, thank you.
Thank you.
I see a question from Tom Rothstein. Is it clear that THT interacts similarly with heterotypic fibrils as it does with homotypic fibrils?
Oh, that is a very interesting question. I think we don't know. I think we assume that THT is a wonderful dye that will report honestly what the amino structures is. But I think you are absolutely right. I mean, there can be differences in their interactions with THT, because I think it is also relevant when we see these dye experiments that we have done. We've used a DCVJ, THT, ANS, and even curcumin. They all show differences, and I'm certain that this is something to do with the selectivity of THT towards homotypic versus heterotypic also is in play. So we don't know how much they will differ, but they could differ. And you're absolutely right. It's a great question.